The information in this module is accurate and complete to the best of our knowledge. All recommendations are made without guarantee on the part of the author or the sponsoring institutions. The author and the sponsoring institutions disclaim any liability in connection with the use of this information.
Welcome. In today's lesson, we will talk about patterns of genome variation focusing on recombination. Recombination is a rearrangement of genetic material, especially by crossing over in chromosomes or by artificial joining of segments of DNA from different individuals or even different organisms. Recombination produces a variety of different allele combinations called haplotypes. A haplotype refers to the combination of allelic states of polymorphisms along the same DNA molecule. In other words, a sequence of alleles on the same chromosome or mitochondrial DNA. These polymorphic sites can include any class of DNA variants, from base substitution to copy number variant. Look at this picture. Note that the top two females have identical genotypes, but the haplotypes are different. So recombination increases haplotype diversity, like in this picture. If there was no recombination, 4 SNPs could yield only 5 haplotypes. But in case if you have free recombination, you can end up with a total of 16 haplotypes. So recombination will increase haplotype diversity, meaning increasing genetic diversity. What is the difference between linkage and linkage disequilibrium? Genetic linkage is described as a tendency of DNA sequence that are close together on a chromosome to be inherited together during the meiosis phase of sexual reproduction. In other words, the closer you are, the more linked you are. Linkage disequilibrium, or LD, is said to occur when two alleles that are found together on the same chromosome are segregating more often than expected if they were segregating at random, as if something would keep the two alleles together on the same haplotype. The haplotype between them would not break despite the distance. Linkage disequilibrium, or LD, is also described statistically as a non-random association of alleles at different loci on the same chromosome. In that sense, loci are said to be in LD, or in linkage disequilibrium, when the tendency of association of their different alleles is higher or lower than what would be expected if the loci were independent and associated randomly. How do you measure linkage and linkage disequilibrium? When genotypes at one locus are independent of genotypes at the other locus, the two loci are in linkage equilibrium. Otherwise, the loci are in linkage disequilibrium. The oldest and the simplest measure of linkage disequilibrium, or LD, is denoted as D. D defines as the difference between the observed frequency of two locus haplotype and its expected frequency. D is also referred to as a coefficient of linkage disequilibrium. Look at the formula, which basically shows you the difference between observed frequency and the expected frequency. The maximum value of D can be assumed to be no more than 0.25, and that can occur when capital A, capital B, and lower a and lowercase b are the only chromosomes present, and each has a frequency of 50%. The minimum value of D, therefore, can be no less than negative 0.25, when capital A, lowercase b, and lowercase a, capital B are the only chromosomes present, each with the frequency of 50%. In addition to statistical methods, recombination can be revealed by direct studies in sperm, DNA, and in pedigrees. Recombination rate, of course, is variant across the genome, and it's higher in places called recombination hotspots. These are regions in which recombination is at elevated rates relative to the neutral expectation. We find these by outside of statistical methods, either by single sperm genotyping, which will reveal the haplotypes because of the haploid nature of sperm. This approach is pretty powerful, but it has a disadvantage, is that it reveals nothing about inter-individual or sex-specific differences in recombination behavior. Alternatively, you can look at the entire population and infer recombination from population studies. This is a more direct approach than to study recombination in families. LD allows imputation of genotypes. Because of LD structure, we can measure frequency of one locus and impute the frequency of another locus because of its association. Imputation is a statistical inference of unobserved genotypes. It's achieved by using a known haplotype in a population, for instance from HapMap or 1000 Genome Project in humans, thereby allowing to test the missing genetic variants for association with trait of interest. Genotype imputation helps in narrowing down the location of causal variants in genome-wide studies. Another problem that comes from genotyping is that sometimes you don't know which allele belongs to which haplotype. So we use phasing, a way of going from genotypes to haplotypes. In a diploid individual, the Grammatik phase represents the original allelic combination that individual receives from each parent. It is therefore a particular association of alleles at different loci on the same chromosome or haplotype. So haplotypes are often starting points for looking for genes that evolve in diseases. Haplotypes are starting point to understanding the pattern of recombination LD across the genome and for construction of gene trees, phylogenies of individual loci in human evolutionary genetics. Once haplotypes are deduced, then we can greatly reduce amount of genotyping. In fact, we can use only tag SNPs, those that can be genotyped to represent the entire haplotypes and save the cost of sequencing. Therefore, haplotypes need to be quickly phased out of the genotypic data. So to deduce haplotype from genotypes, the easiest thing is to use Clark's Parsimony algorithm, which was invented by Dr. Andy Clark. We start with a hypothetical diploid locus containing 8 SNPs, as shown in the picture. Sequencing reveals homozygous and heterozygous genotypes, and the underlying haplotypes are deducible using a parsimony principle. I prepared some review questions at the end of this lecture. What is recombination? What is the evolutionary role of recombination? Can you provide a definition of a haplotype? Can you define linkage disequilibrium, or LD? What are the methods of mapping recombination rates across the genome? Can you explain phasing? How do you use tag SNPs? This concludes today's lesson, Patterns of Genome Variation, focused on recombination. Next, we'll be studying the topic, Surveys of Genomic Variation.